Hello, my name is Dhaivat Joshi. Welcome to my Capstone presentation. With the rise of space travel, and especially private aerospace companies, there is more than one question surrounding the future of the human race in space. Today, I aim to answer one of these questions. How does a space environment affect the human genome? Specifically, what do factors like microgravity, zero gravity, or radiation have to do with gene expression, damage done to DNA, and gene regulation? Let's start with the effect that microgravity has on gene expression. In a 2025 Huang et al. Study, researchers sent human mesenchymal stem or stromal cells, also called HMSCs, to the International Space Station for 1 to 2 weeks. By comparing the gene expression profiles with RNA sequencing data between the HMSCs in space and their identical counterparts on Earth, the researchers found that microgravity played a significant role in the expression of genes involved in cell division and extracellular matrix organization. It's fascinating how just by taking the gravity away, the cells are reprogramming themselves at a transcriptional level. When these same researchers built a gene network from the RNA sequencing data that was collected, they found that one particular gene stood out, CCND1 or cyclin D1. This gene drives the transition from the G1 phase into the S phase in the cell cycle. Interestingly enough, this same gene is also involved in cancer development and interacts with chromatin remodeling proteins. This suggests that cell proliferation and epigenetic regulation are being influenced by microgravity. Microgravity also causes stresses on the mitochondria. A review by Ray et al. Reports that microgravity causes mitochondrial proteins to accumulate and interferes with mitochondrial proteolysis, or the process of breaking down proteins within the mitochondria. This mismatch in proteins within the mitochondria triggers a response to increase the number of signaling molecules like FGF21, which are linked to bone loss and reduced physical function of the human body. This is why NASA makes such a big deal about having astronauts be physically fit before and during their time in space. Working out in space is important because of these changes going on within the cells. Space radiation originating from the galactic cosmic rays and solar events is also a factor that impacts DNA. In the Yuan et al. Study, radiation was found to cause base damage as well as single and double strand breaks in DNA. Cells respond to this damage by activating certain genes responsible for controlling the cell cycle and apoptosis. With repeated exposure, this becomes an important consideration when protecting astronauts from cancer risk and mutations in the future. This is basically 1,000 times riskier version of going to the beach without any sunscreen. Finally, space missions also contribute to effects in the microbiome, metagenomics, and the immune system. Researchers Sun et al. Found microbiome changes that overlap with aging. Astronauts after spaceflight and older members of society similarly have higher levels of IL-6 proteins, upregulated IL-6 related genes, and higher levels of MCP1 proteins. There's more chronic low-grade inflammation present and a less efficient immune response in both astronauts and the elderly. Also, the gut microbiome is also affected. Spaceflight changes which bacteria live in the gut and how active they are. So if anyone's a picky eater, it's not just you being dramatic about food, it's also your gut bacteria. So with all of these factors of space travel affecting the human genome, the question becomes, is it safe and ethical to send people to space for a long duration? Would you still want to travel to space once that becomes commercially available? With the launch of Artemis 2 in a few months, humans will once again be journeying to the moon. With the later missions, the plan is to set up a base on the moon for travel to Mars. How can we ensure that all humans with significant variations in genetics stay safe in space? Thank you for listening, and here are my provided sources.